Hello everyone, we are Group 12. In this video, we are going to talk about ILC1. Our presentation is divided into two sections. Now, let's move on to Section 1. What are ILCs? ILCs are lymphocytes that contribute to earlier innate immune response. ILCs are classified into ILC1, ILC2, and ILC3 based on their cytokines. In this video, we are going to talk about ILC1. How do ILC1 work? 1. Dendritic cells and macrophages eat pathogens. 2. Dendritic cells and macrophages produce cytokines called IL12 and IL18. 3. These cytokines activate ILC1. 4. Activated ILC1 rapidly produces interferon gamma. 5. Interferon gamma activate dendritic cells and macrophages. 6. Increase in MHC class II expression. Activated dendritic cells and macrophages have many MHC class II. MHC class II present antigens to naive CD4 plus T cells. And then many naive T cells differentiate into many effector T cells. As a result, many effector CD4 plus T cells lead to earlier innate immune response. In addition, there are two other reasons for earlier innate immune response. Here is the second reason. Interferon gamma promotes differentiation of naive CD4 plus T cells. Here is the third reason. We'll explain this in further detail. ILC1 have no antigen receptor, which makes it impossible for antigen to activate. ILC1 are activated not by antigen but by IL12, so the specific reaction doesn't occur. As a result, no antigen receptor on ILC1 leads to earlier innate immune response. Here is the summary of Section 1. ILC1 are lymphocytes that contribute to earlier innate immune response. In addition, ILC1 produce interferon gamma and have no antigen receptor. Now, let's move on to Section 2.
Next, we compare ILC1s with natural killer cells. Both ILC1s and natural killer cells don't have antigen receptors and are lymphocytes which are involved in innate immunity. Both cells produce interferon gamma. This makes macrophages active and protect our bodies from foreign matters. If you look so far, you will think that natural killer cells and ILC1s have the same features. What is the difference between natural killer cells and ILC1s? They have two differences. They are whether they have cytotoxicity or not, and where they are. First, about cytotoxicity. ILC1s don't have perforin and granzyme. What are perforin and granzyme? Perforin and granzyme are proteins that are secreted by natural killer cells and cytotoxic T cells. Perforin can make a hole in the membrane of the target cell. Granzyme can invade and destroy the target cell through this hole. In this way, they induce apoptosis and are cytotoxic. ILC1s don't have perforin and granzyme, so they don't have cytotoxicity. Therefore, they don't have a function that kills target cells. Next, about place. Natural killer cells circulate throughout the body, and ILC1s are located on a specific organization from the beginning. Mainly, they are located on the liver and the intestinal tract, which relates with biological defense. We summarize the points. ILC1s are different from natural killer cells in terms of cytotoxicity and place. ILC1s are located on the liver and the intestinal tract. Thank you for watching.